Good morning all, and thank you for attending today. Before I start, just as James said, a general note, as I go through, if you have any questions, please put them in the chat and I'll try to address them at the end. If we don't have time, or if you have questions later in the session, please feel free to send me an email and I'll be happy to talk to you at a separate time. As James mentioned, I'm going to be talking to you today about surface and coatings. So I'll start with a brief introduction to surface and coatings, in particular plasma polymerization, the technique that the group I work with focuses on. After this, I'm going to go through a number of the research and industry linked projects that we're currently working on, and 3 examples in particular. In this case, antimicrobial coatings, antiviral coatings, and also hydrophobic ones. Most of the content is aimed towards providing an overview of some of the work that we're doing around surface and coatings. The seminars themselves were originally intended to be a method of linking in with industry as well. They're provided by the GISMO program, Greater Innovation Materials Optimisation, which I'll briefly cover at the end of the session as well. So if we launch into things, as I'm sure everyone here knows what a surface is, but to be clear, in general going forwards, I'm going to be talking about the top few atomic layers of a material, where your material or your product is interacting with its surroundings. The properties and behaviour of surfaces can be very different to the bulk of the material, and that in turn affects how that material is going to interact with its environment. In many cases, surface properties are essential for your intended use, your function, performance of a material or a product in the way that the bulk isn't. And you can also exploit the properties of a surface to provide different functionality to a material. Maybe you want it to work in a different environment, for instance. Because of all of that, having a good knowledge about a surface, its structure, its chemical makeup, all of that can be very important, and there's a wide range of surface analytical techniques that you can use in that way for research development and also quality control as well. If you do want to impart some kind of property to a material, one of the methods you can do that is by coating it in some way. Functional coatings, so ones that are actually doing something, providing some other kind of functionality to your product or your material, represent a multi-billion-pound industry. They're applied to a diverse range of products, so things from contact lenses to drinks cartons, automotive parts, textiles, packaging, medical implants, and more besides. They're an effective, proven route to add substantial value to existing materials and products. You can use them to enhance multiple material properties. So if you wanted to enhance a material's performance, its reliability, its versatility, maybe you want to use it in a different environment, or even its endurance. And you can impart particular behavior. So if you want something to be specifically hydrophobic, hydrophilic, wettable, adhesive, anti-fouling, or have antimicrobial properties, and so on. Obviously, there are a lot of different coating techniques available. The one I'm going to talk about today is plasma polymerization, which, as I said, is a particular focus of our research group. So in plasma polymerization, you use an organic compound. You introduce that into a vacuum chamber as a vapor, although there are atmospheric variants of the technique. That vapor is then excited with an electric field, the plasma phase. If you put your material to be coated within that plasma, all the surfaces that are exposed to the plasma will be coated. The image here is a lab-based system. So it's based around a central glass pipe in the center. We have a monomer flask at one end. So this is where we have our chemical that we want to turn into a coating. The vapor comes off here into the chamber. We have a pump at the other end, and we have a power supply, in this case, coupled with a copper coil, which excites the plasma inside the glass tube, and your material, which doesn't necessarily have to be sat on the bottom there, it's just to illustrate this, that's coated by the process. You can have a system with a very different configuration or design. You can have things like internal electrodes rather than the external one here, for example, but it should give you an idea of the principle. So plasma polymerization itself has been used since maybe around the 1960s. So things like packaging, automotive headlights, but specifically functional coatings have been looked at for maybe about the last 30 years or so, so early 90s. Key features of the technology are uniformity of the coating, regardless of the substrate, the material that you put into there, or its size. Your coating can be homogeneous, it can be spatially controlled. You can coat a variety of materials, shapes, sizes, geometries. Generally, we say it's substrate agnostic. It's a clean chemical process, solvent-free, and the vacuum process means that you only use very small quantities of reagents within a closed system. The process itself can give you a high value add to your products that you're coating. It has proven scalability. It underpins a range of existing products, lenses, cell culture ware through to juice cartons. So it is proven in scale-up activities. And as expected from the content of this talk, you can use it to create functional coatings. So if you wanted your thing to be hydrophobic, anti-fouling, adhesive, antiviral, antimicrobial, and so on, you can do that with plasma polymerization. So the images on this slide show two of our lab-based systems. The top one is basically the one I showed you a diagram of earlier. It's typical of a lab-based system, whereas the bottom one is something that might be on its way towards a pilot reactor, so something that's much more precisely designed with the ability to add a range of diagnostic tools to it. Going beyond that, you can design reactors to coat runs of hundreds or thousands of items or high-speed roll-to-roll processes, so it is integratable into other manufacturing processes. So having run through some of the basics, I'm now going to start discussing some of our plasma polymerization research projects. So in terms of the work that we do, our aims, plasma and plasma depositions in general, they involve some quite complex processes. Your parameters of the system, how you operate it, those affect both the plasma and the depositions. And important parameters include things like your plasma power, the pressure, the flow rate, your reactor geometry, its design, the power supply, the connections. We personally do a lot of work over different pressure regimes with the goal of developing functional coatings and also advancing control of the growth process itself, trying to retain specific functional groups or larger structural units from our original monomer, original molecules, basically, into the plasma coating. A lot of our work is done with applications in mind. We're always keen to talk to people about different real-world applications, and also we look to test our materials in relevant environments because of that. Overall, our projects fall into broadly 4 categories. So the first is antimicrobial or anti-biofilm coatings, which includes developing plasma coatings with those properties. So we look at things like nitric oxide containing coatings for anti-biofilm. We also look at other antimicrobials deposited from the plasma. But a different approach as well is to use surfaces that might not have an antimicrobial effect themselves, but you can then bind an antimicrobial agent to them. The second project specifically is antiviral coatings. That's actually quite news to us. It's been born out of the pandemic. So if we'd been doing this talk in February, it wouldn't have been on the list. The third set of projects using plasma polymers. So that's for building sensors. So this looks towards medical sensors, things like biomolecule immobilization or nuclear sensors. And then the fourth one, I would say, isn't a specific research project per se, but it covers other work that we do, often with industry. So that's using plasma polymerization for changing intrinsic surface material performance. I'm going to discuss projects within 3 of those categories today. So I'm going to talk about anti-biofilm, antiviral, and hydrophobic coatings to give more of a feel for the kind of coatings that we're working on, we're trying to develop, our methodology and some of the capabilities of the techniques as well. So I'll begin with the anti-biofilm coatings. In this case, it's nitric oxide containing coatings. So I've said anti-biofilm. So as a brief introduction, what are biofilms? Well, they're structured communities of microorganisms growing on a surface of some kind, as opposed to individual microorganisms. Biofilms can have a detrimental effect on a range of sectors, so things like biofouling, water and food contamination, corrosion, through to healthcare, where they have links to persistent infections, antimicrobial resistance, and treatment failure. Although there are positive uses for biofilms in certain sectors, so people look at bioremediation, fuel cells, and that kind of thing as well. They can be very hard to eradicate. They are multi-layered structures. They can often contain different microorganisms. They can be difficult to penetrate with drugs. The biofilm offers more protection against things like pH, so acidity, this kind of thing, temperature, other stresses. So where does nitric oxide come in? Nitric oxide, NO, is involved in the regulation of bacterial processes. It also has wider applications in other biological processes as well. It's previously been shown that nitric oxide can be used as an antibacterial agent. It disrupts a process known as quorum sensing, which is how bacteria regulate cooperative and physiological activities. Essentially, it's them communicating by releasing and sensing and responding to different signal molecules. Nitric oxide has also been shown to inhibit biofilm growth and cause dispersion of formed biofilms. There's been extensive research into nitric oxide releasing polymers, coatings. These things can have limitations though. It can be limited by complexity, cost, delivery mechanism, and also there are concerns around stability and short lifetimes of nitric oxide. An alternative approach, though, to nitric oxide release agents is to use stable nitroxide radicals, which provide properties analogous to nitric oxide without a detrimentally short lifespan. The image here is a live dead stain example from the literature using Tempo, which contains a stable nitroxyl radical, stable free radical, and you can hopefully see the changes in the distribution of the stained bacteria there depending on the conditions used. So if we turn to our work, how we've been working to develop nitroxyl radical polymer coatings, how we've been working to optimize or identify conditions for making our coatings. Work in this section is in collaboration with James Bradley's group at the University of Liverpool. In this case, they have produced the samples and we've done surface analysis on them, but we do make these coatings ourselves as well. This work has been published recently. I believe it was last Friday. So if you want more information, it is available online, or get in touch and I can send a link to you. Here we're working with a stable nitroxyl radical. I mentioned Tempo before. This is the molecule that you can see here. You have your nitroxyl group at the bottom, bordered by 4 methyl groups, which help with the stability. Previous studies in the field have shown that using higher pressure plasma allows retention of fragile moieties from plasma into thin films. And that's basically our approach here. We want to look at how the plasma pressure, the plasma power affect both the plasma itself and the coating that we're depositing out of it. In particular, we're interested in trying to retain this structure of this molecule, and in particular the nitroxyl radical, as that's key for our intended application in preventing biofilm buildup. So the process here was to use a series of different Tempo plasma conditions, in particular a series with varying pressure and static power, and then the opposite, so varying power and static pressure. Our collaborators at Liverpool have also made measurements of the plasma itself around mass spectroscopy, deposition rate, ionic flux. I'm not going to show that information today. It's more of a focus on the coatings themselves. The coatings were deposited onto silicon and aluminium substrates. I will admit they aren't likely to be real-world substrates that you'd want for an application, but for initial studies, they provide an ideal surface for us to analyze. And we used a process known as X-ray photoelectron spectroscopy to do the majority of this work, to look at the surface chemistry. I'll talk about that over the next few slides. The reactor that you can see here in the image, so the photo at the top, the diagram at the bottom, that's the system at Liverpool that was used for the work on Tempo here. That's the setup they used to measure the plasma conditions and also deposit the films in. And again, it shows you some of the differences lab to lab in these reactors. So I mentioned X-ray photoelectron spectroscopy a minute ago, or XPS for short. It's a major surface analysis technique that allows us to determine elemental composition and also chemical state information for contamination. Obviously, other surface analysis techniques are available, and as ever, all techniques have different capabilities, pros and cons. This one happens to be incredibly useful for the kind of work that we're doing. Essentially, it works by shining X-rays onto a sample. Energy is absorbed and electrons are emitted, which themselves have a characteristic energy based on the element they've come from and its local binding conditions. So its neighboring atoms, its oxidative state, it's surface sensitive, and it's quantitative. You can see some examples of Tempo plasma polymer spectra, so the data on the bottom of the screen here. I'm going to go through that over the next few slides and explain the kind of information that we can get out of this technique. So as to the Tempo data itself, what it means, as a broad point with this particular project, we're interested in how much of the structure of the molecule is retained from before, and in particular, that NO bond. The data here on the right-hand side, this graph here, that shows us a number of things. It allows us to identify the elements that we can see in the top few layers of our sample. In this case, it's carbon, nitrogen, and oxygen, as we'd expect, because that's the makeup of our molecule. We can't see any clear signs of our substrate, basically the material we're coating onto, which was aluminium in this case. We can't see any clear evidence of contamination. So this means that our coating contains pretty much what we'd want or what we'd expect. We can also use that data to work out the atomic composition of the top few layers of the coating, and that's displayed on the other side of the screen. As I mentioned earlier, we have two series, one of them pressure constant, varying input power, the other one power constant, varying pressure. We don't see a great deal of change in either of those series for atomic concentration, certainly nothing significant, no clear trends. We do have more nitrogen and oxygen in our coating than the structure of the initial molecule, but that's probably to be expected. We're likely to have post-process oxidation. We may be losing things like the methyl groups and so on in the plasma itself. If we want to look at the effects of power and pressure on the retention of that actual NO bond itself, and also the overall structure, we can do higher resolution scans. Essentially, we're focusing in on those peaks from the previous image with our XPS, and we can start to fit bonding environments to that, basically saying what are the atoms bound to, what are the elements bound to on the surface. This slide shows you the effects of power on our coating. It's the original data. It's way too detailed to run through here thoroughly, but I will give you the broad trends today. So the things we're interested in the carbon spectra, which is this part in the center here, are the carbon nitrogen and the carbon oxygen environments. And on the nitrogen spectra, we're interested in the nitrogen oxygen environment in particular. The carbon peak shape is roughly the same across all our samples, but as power increases, we see a slight decrease in our carbon nitrogen environment. We see a slight increase in the carbon oxygen species. Overall, it suggests we might have more ring cleavage, so this ring structure may be broken up a bit more at higher power. Our behaviour is much more pronounced in the nitrogen spectra. As the power increases, we get less of the nitrogen-oxygen environments, and we get more nitrogen-carbon environments. So that's basically going the wrong way for us. So that's indicating to us that it's probably better for us to use lower power. So lower power is leading to an increased retention of that nitrogen-oxygen bond and the overall structure. If we turn to the effects of pressure, we can see similar changes. Again, this is original data, it's slightly different presentation. Here are carbon spectra, again, towards the center of the screen. It's relatively similar between our pressure ranges. We do have a slight introduction of extra carbon oxygen species at high pressure. The nitrogen spectra is much more clear again. At higher pressure, we have a much higher percentage of nitrogen and oxygen. At lower pressure, we see less of that. We see more of an introduction of other nitrogen carbon environments, so less of what we're after. So again, this gives us a trend that we can look at. Overall, this indicates that higher pressure leads to increased retention of that nitrogen-oxygen bond and the overall structure. So between those two series, we can say our data indicates that higher pressure, lower power allow us to increase retention of that nitrogen-oxygen bond and likely the overall structure. And that's what we're aiming for the particular application in mind. So to summarize this section, the nitroxyl radical or NO-containing coating can be used for anti-biofilm purposes. That's been shown by others in the literature previously. We've been working with Tempi, which contains a stable nitroxyl radical, to deposit coatings with anti-biofilm purposes in mind. We can use surface analysis tools to identify the composition of our coatings. We can identify trends in our coating processes, which help us optimize the process. They help us suggest the best conditions. I should note that those pressure and power trends correlate with the plasma diagnostics that were performed at Liverpool, their measurements of the plasma itself. And the same kind of relationship has been shown with other chemicals. So that's the higher pressure, lower power can lead to greater functional group tension in our coatings. As to where that particular project's going next, well, we do have further studies to do, but broadly, we want to move it on to real world materials, so not on silicon or aluminium, for example. And quite crucially for that kind of application, test against microorganisms as well. So the second section is around antiviral coatings. I did mention this is a new area to us. It's come out of the pandemic. By this point, I am sure that we're all becoming quite knowledgeable about SARS-CoV-2, COVID-19, and viruses in general. But as a general introduction, SARS-CoV-2 is highly contagious. It spreads through multiple routes. The transmission of viruses and indeed other healthcare-associated infections is well documented. You can have routes through spreading infectious droplets through the air, person-to-person contact, or transmission through contact with contaminated surfaces, which are also known as fomites. The quantity of virus on a surface, ability, its resistance against biocidal agents, and actually the minimum infective dose itself are all key to transmission. Things like PPE and social distancing are effective for countering direct transmission. I think that's probably well proven by this point. But transmission via contamination of surfaces does represent a major concern. Even with the introduction of vaccines going forwards, this isn't an issue that's going to go away overnight. So how do we go about reducing surface-based transmission? Well, currently a big thing in probably every workplace is disinfection and cleaning of every surface. We know that there are cleaning agents, alcoholic wipes, other chemicals and so on that are effective against SARS-CoV-2 and also other viruses as well. We also know that cleaning every surface is impractical though, and in certain circumstances it may be impossible. You might miss things. There are materials that are difficult to clean or impossible to do. If you think about materials like fabrics or porous surfaces, it's very difficult to clean those thoroughly. Ideally, you want to have a surface that will kill your virus on contact, deactivate it on contact. So your virus ends up on the surface and it's quickly deactivated. You don't need to do anything else yourself afterwards. So obviously the goal here is to develop surfaces or coatings that can be used on a wide range of materials and have antiviral properties. So we would normally say they're contact killing surfaces. Obviously, we're not the only people to have thought about this, though. There are existing approaches. So there's metal coatings, nanomaterials, paints, dips, and so on. Other coating technologies are available. They have been shown to be effective at killing a variety of different viruses. In any case, any coating technology is going to have advantages and disadvantages. I'm not here to rubbish someone else's solution. Limitations can be material or substrate dependent. They can be chemistry dependent. They can be application dependent. You might have something that's light activated, so it works in a well lit situation, but not quite as well in a dark situation, or more than that as well. In this context, plasma offers us a number of advantages. As I mentioned earlier, you can coat pretty much any substrate. You can coat objects with complex geometry. It's very suitable for textures, for porous materials, basically where the plasma can get in amongst the material because it's immersive. Some other methods may have issues in those sectors. This is ongoing work. I'm afraid I can't mention the specific chemistries that we're using for our antiviral coatings at present, past the point that they are based on proven antiviral chemistries. The idea was to take something that we know should be antiviral, use it in a plasma polymer coating, establish its antiviral in that state, and then use the strengths of the technique to apply it to a range of different applications. Currently, we've made a number of different coatings. We've used surface analysis techniques, such as XPS that I showed you a minute ago, to find optimum coating conditions for our reactor to retain the functional groups that we believe are going to have an effect. We've also tested those for stability. The next step is clearly to say, are they actually antiviral? And we're doing that in partnership with virologists led by Dr. Mohammed Munir here at Lancaster. So I'll quickly run through the process used for testing against SARS-CoV-2 at present. It's based on a Plaque Assay. It's very similar to ISO protocols for other antiviral testing, though to my knowledge, there isn't an official specific anti-SARS-CoV-2 assay. I should note I am not a virologist. This work is done by virologists in category 3 labs. I'm just going to give you a broad overview of the process used at present for this particular test. So the first stage is to take your antiviral coated surface and treat it with the virus. And this is done by applying a volume of SARS-CoV-2 solution, which will have a defined number of virus particles, or by applying a virus-free control solution onto the surface of our pre-coated sample. A second sample that's been coated is placed on top to sandwich the virus between our antiviral surfaces and to increase the chance of contact between the virus and the surface. To make sure we know that if it is antiviral, it's actually going to be that have an effect. This is left for a specific amount of time. That's determined by our study. I've put 30 minutes here. You can have shorter ones or longer ones, dependent on the data you're looking for. After the desired amount of time has elapsed, the surfaces are separated and washed thoroughly into solution. That's the virus preparation, which can then be used for the Plaque Assay. The idea is that if we have an antiviral coating on the surface, there should be less viable virus particles that have survived that solution. They should have been inactivated. Whereas if our surface is not antiviral, we would expect a large quantity, or a larger quantity, I should say, of viable virus particles to survive into that solution. And then when you do your assay, you'd expect to see a difference. So the Plaque Assay itself is fairly standard. A number of consecutive serial dilutions are made. Cells are seeded onto a well plate for testing against. The virus is added to that ready cell plate. It's allowed to absorb. An overlay media is added. It's incubated for 72 hours. And then the plate is fixed with formaldehyde to inactivate any remaining virus to make sure that it's not going to continue to kill cells while we're doing basically the counting step. It's stained using something like crystal violet. Basically, that stains the live cells, the areas where the virus has not had an effect. So if we had active virus in our original solution, it would lyse the cells, it'd kill the cells, so they would not be stained. And you could get unstained patches, or plaques, that can be counted to determine the viral titer in terms of plaque forming units per milliliter. And that can then be compared to your other coated samples, and you can control samples. So here are some photos of some recent runs with some of our samples. I've cut them down to the 3 most concentrated solutions, as it's easier to see the difference. It is performed in a 6-well plate with all 6 dilutions though. Here the purple or purple-blue regions are the stained cells, the live cells. The white or rather transparent regions, spots, are virus plaques, basically the areas where the cells have been lysed by the virus. The first row is the stock virus solution from before. That's the virus solution that we had originally. The next row down is the first dilution, and then the one below is a further dilution, and so on. Now, the reason we've done this basically is it makes it much easier to tell the difference between our samples and allow the plaques to be counted. If there are too many, it might be impossible. So you can see in the top row, first column on the virus control, it would be incredibly difficult to count the white patches on that particular sample, if not impossible. Whereas the next row down, that's actually an achievable task. You can see a difference on this. So I think in general, I'd say focus on the center row, the 1 log 10 dilution. You should be able to see differences in that. So the columns, the first one, are virus control. That's the virus solution directly put onto the cells, so not using our samples. Virus plaques here, so these white spots. The next column is the cell control. So this is basically without the virus. So as expected, we can't see any white spots. There are no signs of the virus there. The third column is the negative control. That's actually a plasma polymer coating that we expected to have no inherent antiviral properties. And here you can see a large number of virus plaques again. It's very similar actually to our virus control. And basically that's saying that our surface isn't having an effect, or if it is, it's a very minimal one. And that's as we'd expect. The last column is one of our candidate antiviral plasma coatings. And hopefully you can see quite a bit of a difference between that, the negative control, and the virus control. You can see plaques present, but nowhere near as many as in the virus control, the negative control. That's very promising. It's basically showing that our surfaces are clearly having an effect. Now, I should note that the virus concentration used here is very high. It's much higher than one would likely to be spread by, say, someone coughing on something. So in a realistic situation, you'd likely see much less on our surface. So to summarize, we've been working to develop contact-killing antiviral coatings, specifically tested against SARS-CoV-2 using chemistries that we know should be antiviral. Our initial coatings are showing promise, viral behaviour. We do have a lot more real-world application work around contact times and different temperatures to look at, as well as testing onto suitable materials, real-world surfaces. We're always happy to talk to industry about particular issues they foresee or they might be having, and discuss whether our coatings might be suitable for their applications as well. So finally, I'm going to cover one more area. Before I mention we do wider work than antiviral and antimicrobial coatings. This is an example. It's development of patented hydrophobic coatings. So this example is from some work that we've been doing with an industry partner under an NDA. So it's very light on specifics. I'm not showing data from any of their products, but basically showing you the process going forwards. And it provides a good example of the utilization of plasma polymer coatings, in this case to produce patented hydrophobic coatings. It does tie back to our previous work though. You have potential situations where you might want a surface to have multiple properties if you want it to be hydrophobic and antiviral, for instance. So you might have a real world setting where splashes occur, so bodily fluids and so on, where this would be an advantage. At the bottom, you can see some examples of steel and glass substrates before and after plasma coating. On the left, a contact angle example where you can see how a drop of water sits on a coated and an uncoated steel substrate. And on the right, 2 glass slides. The rear one is coated with the droplet of water beading on top, and the front one is uncoated, where the surface is clearly much more hydrophilic. I should say that the development of hydrophobics isn't something that's really new, but the specific applications may be, and we may be doing it in different ways to other people as well. So with this particular piece of work, we were given existing substrates, we were given alternate products. As before, our coatings development is optimized by surface analysis. This case is no different, so using XPS again, but also doing elemental mapping of coatings, taking contact angle measurements as it's supposed to be hydrophobic. Performing all of this kind of work allows us to benchmark against requirements or alternate products. It gives us a point of reference for developing prototype coatings. Basically, it tells us what it needs to do under our lab conditions, as well as what our partners' requirements are. And the approach here is probably quite familiar by now. We use plasma polymerization to coat a surface with a monomer, with a molecule out of the plasma to give it hydrophobic properties. In this case, we're using pattern masking to provide hydrophobic regions. And the prototype samples are both tested in our lab against our benchmarks that we were talking about a second ago, as well as passing back to the company for real life application testing. So basically expected use conditions. In the case of the pattern masking, we can also use selected area XPS. We can use elemental mapping to make sure we're taking our data from the correct areas of a sample. Now these plasma polymers, quite often you can't see them by eye. So if we had a pattern there, I might not be able to see it myself or under microscope. So having some kind of technique where we can map the areas that we've patterned is incredibly useful. In this case, the purple image at the bottom corner is an elemental map, which shows us clearly different regions. So you've got a kind of central circle there where we've got a lower signal and the outside edge signal. So those correspond to uncoated region in the center, coated on the outsides. And by taking our XPS data from within or outside those regions, we can see a clear difference in the surface chemistry, the binding conditions. So I put two plots on the right-hand side. The green lines are the coated areas. So this is where we've got the hydrophobic coating, this top one here, and this top one here. And the red are the uncoated regions, the etched regions. So the bottom line in both cases in red. You can see quite a change in the elemental concentration. So in the top picture, the positions of the peak, the intensities of the peaks, and also a lot of difference in the carbon spectra. So this is zooming in on one of those and actually looking what's binding together. So the different peaks there indicate different carbon binding environments. One of the key things I haven't mentioned so far is the need when making application-based coatings of making sure your coating is stable under expected use conditions. So by that, I mean if you expect a surface to be used with certain chemicals, or if you want it to be used in a specific environment, it needs to retain its function for a suitable length of time within that environment or within that process. You don't want something breaking down partway through a process, particularly if you want to sell it to someone. If you want it to do something that you've said it's going to do. And you might need something different for multiple use items rather than single use items as well. In this case, we're testing hydrophobic coatings against common solvents. A quick way of checking this in the lab is by soaking in solutions for various amounts of time, or by performing wipe tests with solvent-soaked wipes. The figures here show the effect of 5 common solvents on a coated glass slide. And on the other side, in this dotted box, it's just showing the difference between an uncoated glass slide and a coated glass slide. All those measurements are single static contact angle measurements, but there is no clear significant change in angle for any of the solvent-soaked solutions, sorry, solvent-soaked samples rather, which provides evidence towards the coatings being likely to be usable in light solvent use conditions. They're not breaking down. So to summarize this section, aside from specific antimicrobial and antiviral coatings, we also work on wider coatings for modifying material surface properties. An example of this with an industry partner is hydrophobic coatings, but that could be other things as well. If you want something to bind something specific, maybe it's biomolecules. If you want something to be adhesive or have any other properties, that falls into this section as well. Our samples, again, were coated by plasma polymerization, but we used pattern masking to make hydrophobic and hydrophilic regions. Again, surface analysis techniques were highly useful for surface development, R&D, quality control. Here we've also used them for clear identification of our patterned coating chemistry as well. And when looking at applications, stability testing can be vital. You would probably do much more besides this as well. You might do things like storing something for several months and then retesting it later as well, to make sure that your coating lasts for a suitable period of time in various conditions. But in general, you want your coating to work in the intended environment or application. So to summarize overall, hopefully this talk's given you a bit of a window into the types of techniques, the methodologies we use, the research that we're doing, and hopefully it sparked some interest. General points: surfaces are critically important in how a material interacts with its surroundings. Functional coatings are an effective, proven industrial route to add substantial value to a product. They can be used to enhance multiple material properties. Plasma polymerization is an effective method to coat a range of materials and also introduce functionality. And surface analysis can be key to aid coating R&D quality control. I've also given an overview of some of our research, some of our projects in 3 areas. So anti-biofilm coatings, antiviral coatings, and plasma polymers for changing intrinsic material performance, in this case, hydrophobic coatings. Before we finish today, I need to talk briefly about GISMO, the Greater Innovation for Smarter Materials Optimisation project, which has provided the opportunity to do these seminars. So GISMO itself is a European Regional Development Fund programme. It's designed to help businesses with R&D, help develop new products, processes, and help understand ideas or different techniques, to access facilities and technology at Lancaster University, including the things I've talked about today, to help build relationships, projects, or collaborations with academics. GISMO links into Lancaster University's Material Science Institute as well, which ties around 200 researchers across physics, chemistry, and engineering, but also links into biologists, medical researchers, social scientists, computer scientists, and more. The project itself is based in Cheshire and Warrington. It provides eligible SMEs fully funded access to resources and facilities at Lancaster in the form of de minimis state aid. And GISMO itself is based around 3 main themes which tie to research strengths at Lancaster. So additive manufacturing, chemicals and hydrogen, and surface and coatings. And as you can probably imagine, I work in the latter area. If you are a company in Cheshire and Warrington, we'd be delighted to talk to you more about this opportunity. If you work somewhere else, please still get in touch. There are other mechanisms, there are other projects that we might be able to work with you on. It's just we have this specific opportunity to work with Cheshire and Warrington-based businesses via this programme. We also have another seminar coming up early next year. The plan is to continue them going forwards. I believe every month or 2, our GISMO team are working out a schedule at present, and topics will tie into the 3 themes I mentioned earlier, so additive manufacturing, chemicals and hydrogen, surface and coatings. It's not all going to be surface and coatings based, but our next seminar will be. And that's going to be given by Professor Rob Short, who's the director of the Material Science Institute, on Friday, the 5th of February. And he's going to be talking about life science and biomedical applications for coatings technology. Finally, I'd like to acknowledge the support of a number of people at Lancaster and Liverpool for their work, as well as our funders. EPSRC funded the majority of the work that I've talked about in this seminar. But I'd also like to acknowledge the support of our GISMO team, the ERDF programme and the Northern Powerhouse that support GISMO, and a number of other projects at Lancaster University. So thank you for attending. If you do want to know more about GISMO or any of this work, please visit the GISMO website or email either myself or the GISMO team. If there are any questions, I'm going to do my best to answer them in the remaining time. If we don't get through them, or if you have them later, please feel free to get in touch later as well. Thank you.
Thanks, Alex. So we do have one question from Stuart. Are tempoplasma polymer coatings capable of being used as a base for treating porcelain patios to prevent growth such as mold and bacteria?
Right. I think that might be something that's slightly difficult to do. You might be able to. You can definitely coat, various different surfaces. You would be able to coat the material. If you're talking about full-size sort of patio slabs, that kind of stuff, it might not be the best process to do it. I think that might be slightly difficult to integrate. There might be a better way of doing it, but you could potentially use some kind of coating that contains tempo or something else analogous to that. I'm not saying no, but I suspect there might be an easier way to do that for things like, I suppose, porcelain slabs.
Got another question from Stephen.
Mm-hmm.
Are ALD facilities available, or can the facilities at Liverpool be included as part of the project?
So there is a range of facilities available. ALD, off the top of my head, there may be a system. I would have to check. I know there was a bid for atomic layer deposition at some point, but I would have to check and get back to you. So maybe if you drop me an email, I can follow up on that one. As to facilities out of the universities, well, the GISMO program itself covers Lancaster University, so we wouldn't be able to do, basically, we wouldn't be able to provide you access to the facilities at Liverpool as part of the program, but there might be some kind of collaboration in a different way that could link that kind of stuff up. And I've been told by someone else, yes, there is a, thank you Manus for that, there is a new atomic layer deposition system, so there would be a potential to do work around that.
Okay, we've got another question from Sridhara. Have you tried plasma polymerization on other cured polymer coatings, e.g. PU? And do you see them working together for effective antimicrobial properties?
So I haven't personally tried putting it on top of a different polymer coating. We've put it on things like plastics, including polyurethane before. I've not specifically tried integrating it with a different polymer coating process. I believe you could do. I mean, you can put pretty much anything into your plasma reactor and coat it. So there could well be strengths involved there. So maybe if you get a base layer with your other coating process first and then use the plasma polymer to give it a different functionality as well, that might be something that's quite interesting to look at. I think it's something that would need to be discussed or thought of over more length, though, potentially.
Thanks, Alex. If there are no other questions, we've got a question from Manus. Can the plasma deposition be used on Fluoro Elastomers?
Well, you should be able to. I mean, basically, so long as you can put what you want to be coated into the vacuum environment, you can coat it. So yes.
Question from Simon. Is the system a possible replacement for galvanizing steel as an anti-corrosive method?
You can coat steel. You can make anti-corrosive coatings, I believe, as well. So potentially it will probably depend on your application. I mean, plasma coatings, you can make things that, say, are much more oily, or you can make things that are sort of hard glass-like coatings, or anywhere in between. It might depend on what you want to do with that steel later, though. So if you wanted to put it into an incredibly hard wear environment, we might be able to do a coating for it, but it's whether you'd also have maybe exactly the functional behavior you want as well. There might be other techniques such as galvanizing that are easier to do or maybe slightly more suitable for that purpose. But lots of anti-corrosion work has been done before. on a variety of different substrates. I think Rob's mentioned that in the chat. So thank you for that, Rob. So it's a sort of a yes, you can, but it might not be perfect for that particular application. It just depends what you want to do with the steel later, probably.
Okay, I'll just give it a bit in case there are any more questions. If you do have any questions after this session, of course, you can email Alex, or if you have a broader question, you can get in touch with us via the GISMO inquiries email. And after this session, we will also be in contact with a survey, which we'd be very grateful for you to fill in. So if there are no more questions, I'll hand back over to James.
Thank you, Jenny. And thank you too to Alex. I'm sure you'll all join me in giving a virtual round of applause to Alex for that insight into surfaces and coatings and plasma polymerization particularly. Thank you so much for joining us today. We hope that you've learned something from the seminar. If you do have any further questions or you'd like to talk to Lancaster University about involvement in the GISMO project or in our wider offer, please do get in contact with us through either Alex's email address that you can see on the screen there or the GISMO email address, and do have a look at our website as well. We hope that you'll join us again for the next seminar on the 5th of February. But thank you again for joining us today, and I hope you have the rest of a good day. Goodbye now.